Looking at the surface of a cell membrane, you immediately notice its spatial complexity. It is an undulating, highly organized barrier, packed with distinct interlocking structures that govern everything moving in and out of the cell. Yet, despite the dense visual variety of these organelles, the entire cellular environment is constructed from a remarkably narrow inventory. They are predominantly built from just 4 major classes of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. The cell achieves this complexity through modularity. Massive, intricate macromolecules are assembled simply by snapping together smaller, foundational units called monomers. At the atomic level, the ingredients are even simpler. Carbohydrates, lipids, and proteins are all built from variations of the exact same primary atoms: carbon, hydrogen, and oxygen. Because the atomic ingredients are nearly identical, a molecule's biological function is dictated almost entirely by its precise 3-dimensional geometry. The specific angles, kinks, and exposed functional groups determine exactly how a molecule will behave inside a living system. To understand the physical limits and functions of living cells, we have to decode the geometry of these monomers. We start with carbohydrates. Chemically, these are aldehyde and ketone derivatives of polyhydric alcohols, all built onto a standard stable carbon backbone. This 2D Fischer projection maps the straight chain form of D-glucose. You can see the carbon backbone running down the center. At the top, carbon-1 forms an aldehyde group. Down near the bottom, carbon-5 holds a specific hydroxyl group. These carbons are chiral centers. When an alpha carbon is bonded to 4 different groups, the molecule becomes asymmetric. This allows it to exist as distinct spatial stereoisomers, mirror images of the exact same chemical formula. Depending on whether that carbon-5 hydroxyl group points to the right or the left, you get either the D isomer or the L isomer of glucose. Biological systems are highly specific, almost exclusively utilizing D sugars for their cellular processes. In the aqueous environment of a cell, glucose rarely stays in a straight chain. It undergoes hemiacetal formation. The chain bends, and the hydroxyl group at carbon-5 reacts directly with the carbonyl group at carbon-1, closing the molecule into a cyclic ring. That specific ring closure converts the former carbonyl carbon into a brand new chiral center. We designate this the anomeric carbon. This cyclic diagram illustrates mutarotation. In an aqueous solution, the ring rapidly breaks open into its linear form and snaps shut again. As it does, the hydroxyl group on the anomeric carbon spontaneously switches between the alpha position, below, and the beta position, above. Chirality also creates epimers. If you take glucose and flip the configuration of the hydroxyl group at just carbon-2, you create mannose. If you leave carbon-2 alone, but flip the hydroxyl at carbon-4, you create galactose. These microscopic geometric shifts dictate biological compatibility. Enzymes and receptor proteins operate on exact physical fits. A flipped bond determines whether an enzyme can bind to the sugar or if it bounces off entirely. A single flipped hydroxyl group redefines the molecule's identity, its name, and its biological potential within the cell. Cells rarely rely on isolated monosaccharides. They link these single units together to form oligosaccharides, and eventually massive polymer chains called polysaccharides. This animation shows the mechanism used to build those chains: dehydration synthesis. The hydroxyl group of one sugar merges with the anomeric carbon of an adjacent sugar, releasing a water molecule in the process. What remains is a covalent oxygen bridge called a glycosidic linkage, securely locking the 2 monomers together. If you join a specific D-galactose residue to a D-glucose residue with this exact linkage, you produce lactose, a common oligosaccharide. The orientation of this connection, whether the linkage forms in the alpha position or the beta position, creates drastically different macro properties for the resulting polymer. This model represents starch. When a chain is built entirely using alpha D-glucose linkages, the molecule naturally curves. It forms a loose, accessible coiling structure. This open geometry makes it easy for enzymes to snip off glucose units, making starch highly efficient for energy storage. But if the cell builds a polymer using beta D-glucose linkages, the chain remains perfectly linear. These straight chains pack tightly together, forming extensive hydrogen-bonded matrices. This is cellulose. The tight geometric packing makes it rigid, highly stable, and completely indigestible to most enzymes. Chitin operates on the same logic. It uses a beta linkage but replaces one hydroxyl group with an acetamino group. This extensive cross-linking makes the polymer tough enough to form the exoskeletons of insects. The cell's physical choice between an alpha or a beta linkage at the monomer level directly dictates whether the resulting macromolecule functions as metabolic fuel or structural armor. This reliance on shape extends to lipids. These are highly hydrophobic molecules, primarily assembled by linking long hydrocarbon chains to a glycerol molecule via an ester linkage. Looking at this diagram, we can see the physical impact of hydrocarbon saturation. The saturated fatty acid on the left contains no double bonds. Its carbon atoms are fully saturated with hydrogen, resulting in a straight, rigid structure that can pack tightly together. The polyunsaturated fatty acid on the right behaves differently. Every double bond in the chain removes hydrogen atoms and introduces a severe, rigid kink into the molecular geometry, forcing the lipid tails apart. We map this geometry using a strict nomenclature. A designation like 18:2 delta 912 tells us the chain is exactly 18 carbons long and contains exactly 2 double bonds, located strictly between carbons 9 and 10 and carbons 12 and 13. Simple lipids, like triosoglycerols, are tightly packed and stored for energy. But compound lipids, like phospholipids, use their specific kinked geometries to spontaneously assemble into the fluid barriers of the cell membrane. To regulate that fluidity, the cell embeds sterols, like cholesterol. These lodge themselves directly between the lipid tails to physically govern how permeable the membrane is. Finally, we apply this logic to amino acids, the fundamental monomers that assemble into complex proteins. Despite their vast array of functions, all amino acids share the same overarching chemical scaffold. This diagram shows that shared anatomy. At the center is a chiral alpha carbon. It is bonded to a basic amino group on one side, an acidic carboxyl group on the other, a standard hydrogen atom, and a fourth variable position called the R group. That variable R group is the only component that changes. It can be a single hydrogen atom, a methyl group, or a complex ring. That specific geometric and chemical sidechain provides the unique identity for all 20 standard amino acids. Just as sugar linkages govern a cell's rigidity, the specific geometry of lipid kinks and amino R groups governs the fluidity and the reactive machinery of the entire cell. As we zoom out from a single molecule, we see how these components integrate to build the operating plasma membrane. The hydrophobic lipid tails pack together, utilizing their precise degrees of saturation to maintain a physical, impermeable boundary around the cell. Attached to the surface proteins and lipids are glycoconjugates. These carbohydrate chains project outward into the cellular environment, acting as highly specific informational arrays for cell-to-cell communication. These massive, complex macrostructures are the direct and unavoidable result of the stereochemical rules governing their monomers. The architecture of the cell functions entirely within strict physical limits. A slight geometric constraint, whether it is the mutarotation of a glucose ring or the epimerization of a hydroxyl group, determines what structures can physically be built. Life is an emergent property of these exact molecular angles and covalent bonds. If you want to understand how a cell operates, you must look at its chemical geometry.